So we're very happy. We have today Pau to explain to us, to talk about pseudobulk analysis and differential express genes, also mentioning his tool, Decoupler. Did I pronounce it correctly?
It's a hard name. It's a decoupler. You can pronounce it however.
Decoupler. Very good. I have been writing it for the whole week, but pronouncing it is not my forte. So yeah, we're very happy to have you. Thank you so much, and you can start.
Okay.
You can share your screen.
Let me share. So do you see my screen?
Yes, I think you should, put full screen or something.
Yeah.
Is it?
I think if I press here,
yep. Yeah.
It should work, or there should be also a presentation. Let me present a slideshow.
Yeah.
Perfect. Then let's make this smaller. Okay.
Yep. Perfect.
So thank you for having me. I'm Pau. I'm a PhD student at Julio Sáez Rodriguez Group here at Heidelberg University Hospital. And today I'm going to talk about enrichment analysis, but at the pseudobulk resolution using the tool that we have kind of recently developed called Decoupler. There's a little bit of an introduction. So in our group, as in many others, we basically get omics data from healthy and diseased patients. And our objective in these projects is to try to characterize the molecular response that happens in disease, right? And we can do this by applying classic stats and, for example, running differential expression analysis to identify which are the genes or which features are changing in disease compared to healthy. However, this is quite hard to interpret because just, for example, with differential expression analysis, we might get more than 1,000 genes, and it's really hard to come up with a mechanistic interpretation of this. So in our group, we specialize in enrichment analysis. So basically, we use prior knowledge. You can understand prior knowledge as gene sets. So for example, we know that the transcription factor targets a specific collection of downstream target genes, or that some genes are associated to a given pathway and so on. So basically what we do is we compute these enrichment scores. And with those, we lower the dimensionality of the problem, which increases the statistical power and also provides a mechanistic interpretation of our data. So to perform enrichment analysis, in the group, we have developed this tool called Decoupler, which is just a unified framework of several enrichment methods that have been going around for a while already, and a collection of utility functions to process single-cell data, bulk, and pseudobulk profiles. It's available both in R and Python as separate implementations, so don't worry, you don't need to use Reticulate or try to battle different environments. And it has access to many geneset databases through the OmniPath database that we have in the group. Some of the enrichment methods you may know, for example, geneset enrichment analysis, representation analysis, AUCell, and so on. But basically the idea is that you come with your omics data, your prior knowledge resource, which you can understand this as genesets, you pick your enrichment method, and then you get your enrichment scores, right? So enrichment analysis can be done at the single cell level. And for example, it can help at cell type annotation. Here we have the classic PBMC. And for example, if you have marker genes for natural killer cells, we can perform enrichment analysis and we see, oh, surprise, cluster 4 seems to be enriched by natural killer cell markers, right? And here we would annotate this cluster as being natural killer. If you have information for transcription factor targets, we can estimate transcription factor activity. For example, here we see this cluster of B cells has a higher activity for or enrichment scores for PAX5. And as you may know, PAX5 is a transcription factor that is necessary for B cell maintenance. So this is kind of expected to see this, but it's also nice to see. We can also estimate pathway enrichment scores here just to show that this can also be applied to spatial data. So here we do it on a Visium slide. Here we have a Visium slide with different clusterings. And we see that, for example, this cyan cluster seems to be enriched by interferon alpha response, or there are different spatial distributions of these G2M checkpoint, right? But so enrichment analysis can also be done at the contrast level. Basically, this means that so we have healthy and diseased samples, right? And we perform differential expression analysis, so we know which genes are up or down in disease. And from the results of this test, we take the gene level statistics and we perform enrichment analysis on those. And for example, here we see which transcription factors are predicted to be active or inactive in a bulk data set. We have a vignette showing how to do this in bulk, and also at the pathway level. But yeah, coming back to single cell. So obviously we can apply, so we can perform differential expression analysis at the single cell level, but it has some limitations. Because we assume that each single cell is a sample, we will get inflated p-values. Also, because cells coming from the same patients or samples share a little bit of noise. So we have this sample batch effect. This will affect the results of differential testing. And also because each sample can provide a different amount of cells. All of these are biases that will make a lot of false positives in differential expression testing. So as Lucas and Anna described in the recent review, best practices for single cell analysis across modalities, there are two approaches to mitigate this, and one is to use generalized mixed effect models. But another way simpler approach is to just do pseudobulking of your data. By pseudobulking, so this is what we mean. So nowadays in most atlases, the experimental design is that we have different samples, right? And from each sample, we get a collection of cells. This is their gene expression. And obviously these cells, we cluster and classify them into different cell types. So the idea here is that we summarize these sparse gene expression profiles into rich profiles at the cell type and sample resolution. This summarization, the classic way of doing this is just by summing the counts, the raw integer counts, but in Decoupler we have implemented that you can do the mean or you can do any summarization function that you prefer. But the idea is that you get this cell type per sample richer profiles. So with this, I'm going to go over a use case so we have a vignette showcasing how to actually do this analysis. And for that, we use these blood myeloid cells from healthy and COVID patients. The experimental design is quite easy. So we have 3 controls and 3 infected samples. And yeah, we have different cell types and a good collection of cells and genes, right? So we start by, it's very easy to use. It's literally a one-liner. So we use this get pseudobulk function from Decoupler. And as you can see, we obtained 38 different profiles. So remember, 1 for each cell type and patient. So in Decoupler, we also include quality control plots and filterings once these profiles are obtained. We have at the sample level and at the gene level. For example, at the sample level, here we see so for each pseudobulk profile, we can know from how many cells it was created. And for example, these profiles were created using less than 10 cells. We could think that those are rather noisy, so we might want to just keep these samples, right? And at the gene level, we would like to keep genes that are expressed in at least a number of samples and that have a minimum amount of counts. Actually, this filtering strategy is the same one that EdgeR uses, but yeah, we have re-implemented it in Python and also provided some utility plots to help decide the threshold parameters. Then we apply DSIG2, but the Python version. Finally, there's a nice implementation in Python, so it's really cool that we can stick to do enrichment analysis in Python and not to have to jump back and forth from different languages. But yeah, as you can see, PyDSIG is rather easy to use, so it's a collection of one-liners. In the end, we get the logfile changes and the stats for each gene, and we can visualize this using some Volcano, plotting utility functions. So here is the Volcano plot of the contrast. So then, as I was mentioning, we can actually perform enrichment analysis from the results of differential expression testing. And again, here we show that with a very simple one-liner, we can run this method that it's called univariate linear model. And yeah, we can easily visualize the results. So for example, for T cells in these COVID samples, we see an increase in activity for these transcription factors and a decrease of an activity for these ones. And the same for pathways. Yeah, so obviously, I mean, it's kind of expected. We see an increase in JAK-STAT. Those are T cells. So yeah, we expect some immune response, right? But anyways, so I just want to briefly mention also that, so in Decoupler, we have spent a lot of time working on the documentation. So we have vignette showcasing how to use it for cell type annotation, pathway and transcription factor activity inference. We have a very detailed vignette showcasing how to run functional analysis at the pseudobulk level, spatial level, bulk level. We also provide a benchmarking pipeline because obviously, like, we would like to know how these different enrichment methods work, and we have some benchmarking data where we can, basically evaluate how they work. And we also provide conversion to other organisms. So if you're working with something that is not human, we also provide wrappers to transform your prior knowledge resources into your model organism. And with these are the take-home messages. So Decoupler is a framework to perform enrichment analysis at the single cell or contrast level. Enrichment analysis now is possible fully in Python. It's also, so we spend a lot of time on scalability. So it works in single cell. Differential expression analysis at the single cell resolution has some limitations. And one way to contrast this is to use pseudobulk, which basically summarizes the counts into cell type and sample profiles, providing richer profiles so we don't have so many dropouts. And we have the actual true replicates in that information. And that, basically we have a complete pseudobulking pipeline that can be found at Decoupler's Python documentation. And with this, I would like to thank you for your attention. Especially my lab members for their constant support, and also for the scverse team for letting me speak today, and for the amazing job they are doing for implementing all of these tools into the Python environment. So thanks.
Thank you. Great. Very good presentation. Thank you so much. So I will ask, okay, great. I don't have to ask. I want about to ask to clap you with the animations, but also I will ask to raise your hand if you have any questions. Perfect. So Maren?
Yeah, Pau, thanks for this amazing talk. I struggle with differential expression analysis usually because I always have to go back to R and HR and do stuff. I was wondering how you would integrate random, so like mixed effects. Do you have like a formula setting like in similar to stats models, or how does that work?
So currently, I don't know if PyD6 supports this, although they are actively working on trying to make it as close as the original implementation, but I don't know if it's currently available. So full disclosure, I'm not a developer for PyD6, so I just use it, but
Okay, so I probably have to relay that question to the PyD6 people.
Exactly,
that's totally fine. May I shoot another question? Have you ever looked at, so you can do your differential expression analysis also on like a single cell level, and when you do that, do you see differences based on the cell type abundance? So if you test in a rare cell types, we happen to see, let's say, much more noise than compared to abundant cell types. So, and that the number of genes that you get as a DE are dependent on the number of cells you have per group.
I mean, so there was this benchmark that was actually looking at this. It's this, how is it called? 1 second. So personally, I've never tested it, but so confronting false discoveries in single cell differential expression. I think there they had a look at this, and yeah, basically what they saw, if I remember correctly, is that yeah, working at the single cell level will always increase the number of false positives that you get from differential testing. But again, I'm just a user in this case.
So what would be your recommendation?
Always go to so transform your data to pseudobulk profiles. So then it's easier to actually reuse all these bulk differential testing. It can be Lima, it can be HR or DSIC too, or use these mixed models, but I don't have that much experience with those. And also from what I've heard, they are computationally more intensive because
they are, yeah.
Yeah.
Okay, thank you so much.
Thank you.
Tu, you can go next.
Yes. Can you hear me?
Yes.
I have a question. Usually when I do pseudobulk RNA-seq, I tend to calibrate batch effect by mutual nearest neighbors. So if I put batch as a factor in the design formula, does that mean I don't need to do batch effect correction anymore?
Exactly.
What's your advice?
Exactly. So you can take care of the batch either at the, I mean, you could correct your gene expression levels, but I actually prefer to include the batch at the differential testing level. So these frameworks, they allow for regressing out these batch effects whenever we are testing for differential express genes.
Because I have a feeling, although I don't have proof, this batch effect correction sometimes overcorrect and make some cells look so similar that they should be biologically slightly different. However, they like project them on PCA plots, they look so similar. Because this kind of signal has been removed by batch effect correction. That's why I think.
Here you're talking about cells. Am I getting this right, that you're doing pseudobulk of nearest neighbors? So this would fall more into the meta-cell definition rather than the pseudobulk definition, or?
Yes.
Yeah, so whenever the whole meta-cell definition is rather hard. There's a lot of work. Recently, there was this C-cell paper. So this is completely different because here we actually collapse all cells that belong to a given sample into a single profile. So we get the actual true replicate, but we miss a lot of, so we don't get cell state definition there. I mean, you could do it at a cell state definition, but usually we work at the cell type level.
Let me ask you one quick question. Maybe it's, do you still use p-value then in differential gene expression analysis because you are comparing one sample to another sample?
No, we're comparing groups of samples. So the idea would be that you have an experimental design. So for example, the COVID one that I was showing, where we have 3 patients or samples that come from controls and 3 patients or samples that come from COVID patients. So we actually do a 3 versus 3 test.
Okay. Thank you so much. By the way, very nice presentation.
Thank you.
Arco?
Hi, Pau. Thank you very much for the presentation. It was very interesting. Does Decoupler already offer some of these resources that you use for the prior knowledge out of the box?
Exactly. Those are one-liners, so you can just pull them. And basically, so in the group, we also have a prior knowledge metadatabase that it's called Omnipath. And basically what it does, it processes all these different databases and provides them as data frames. It's available also both in Python and R. But yeah, from Decoupler, you just do DC get _resource, and yeah, you just fetch it from the internet, already processed and ready to use.
Okay, cool. Thanks.
Thank you.
Valentin?
Hey, thanks a lot for developing this tool. I've been enjoying using it.
Thanks.
I've been wondering on some of my analysis when I do differential gene expression, in this case with DSEC2, and do a volcano plot, I see like super strange lines. Have you seen this before? And do you have any idea where this comes from?
So what I've seen is, I think here maybe Konstantin will be better suited to reply, but what I've seen is that this is an effect of the log-fold change shrinkage. That basically, what I see is that, yeah, basically log-fold change get shifted to close to zero. But I don't know what to say about that. Sorry.
Interesting. Yeah, maybe if I can chime in there.
Yeah.
I would have expected that if you have these like kind of lines, like in the volcano plot, now also seen them, that they're just because you have like a discrete number of non-zero values. So all are zero but one, then depending on the estimated variance, I guess you can have these kind of effects. But I don't quite remember. I think I once looked into this, but Paul maybe.
It could be also that you're plotting the actual statistic and not the log-fold change. So if you plot the statistic in the volcano plot, you will get a one-to-one relationship with the statistic and the p-value.
Yeah. I mean, I don't know if it helps. I have an example which I could share quickly, but I've just been wondering if this is an issue or not. So.
No, it's so the thing is that in my opinion, volcano plots are not that informative. And for example, for the actual enrichment, we don't use the log-fold changes. We use the t statistic from that we get from DSEC. Yeah. Why? Because so we like this property where we know that the change is also encoding for the actual significance of the contrast. So, for example, if you get like a huge log-fold change, but we know it's not significant, this will affect your enrichment results afterwards. So it's rather more conservative and safe to just plug in the obtain stat, not the log-fold change, but the stat that you get from DSEC2 for enrichment analysis.
So I'm more asking because I want to understand how it works. It's more like for external analysis. So I personally prefer heat maps and these kinds of things. But when people ask for volcano plots and I cannot explain this pattern, it's like a bit, yeah. But thanks.
Thanks.
Jennifer?
Yeah, thank you for the presentation. I had a question about the cell type annotation. Is it possible to pass custom marker genes in? Okay.
Yes, so basically, as long as you provide, so we have this format where we have a source, and so for each source, which can be a cell type, can be a transcription factor, can be a pathway, you need to have a collection of target genes. So basically a classic gene set. As long as you have that, you can just provide your custom gene sets and perform enrichment analysis on those. Yeah.
Okay. Okay, very good. So it doesn't require sort of like full change difference of a gene between like two cell types?
No, not necessarily. So you can provide weights to that gene set if you want, because we have methods that can actually account for those gene set weights when performing enrichment analysis. But if you don't have, it's okay. You can set the parameter weight equals to none, and that's it. It will run.
Okay. All right. Thank you.
Thank you.
It's my time to ask a question then. Do you know, so you say that it's working on single cells, right?
Yeah.
Do you have any idea how much it could scale in terms of samples or cells? Like, because you said it's actually scaling, and that's super interesting.
Yeah, so obviously, I mean, there's a limit to that.
Yeah.
But so I have, I mean, when I say it's scaling, is that you can run it on your local machine most of the time. So you can perform enrichment analysis at the single cell level. So usually I work with atlases of around 100,000 cells. and it runs on my local machine in, let's say, less than 5-10 minutes. But it also depends on the resource that you're running and the method that you're running and so on. But yeah, for example, this actually, so we have the R version of the decoupler. That's not possible in the R version. Unfortunately, you need to switch to, but it's also, so it's not, so R has some limitations, but it's also a problem of how the R implementation of the decoupler works that could be better. But yeah, currently the fast version of the decoupler is the Python one.
Yeah, okay. But, so, but I guess it's kind of, how can I say, linear, let's say, or exponential timing. If I would go to a server and I would do the same, would it be the time exponential, like, or just linear?
Yeah, I have no idea on this. I'm not a computational scientist by training, so I.
No, it's okay. Don't worry.
Sorry.
Thank you anyway. I mean, I can try. So I will let you know how it goes.
Okay.
Okay. Any more questions? Yes, Lina.
Thanks for the presentation. I have a question regarding the actual generation of the pseudobulk counts. So do you consider, sometimes you have different number of cells between different samples. Do you somehow like take the same number of cells from each sample, or do you assume that the DSeq normalization will correct for any kind of, sequencing depth or cell number differences?
Exactly. So when working with differential expression analysis, we assume that DSeq is gonna, so DSeq normalization is gonna take care of this. But if you want to, for example, I don't know, project your samples into a PCA or a UMAP or whatever, then what we do is we actually just run standard normalization and just log, so scale log. And yeah, and we just work on that. One thing though is that, so the get pseudobulk function also provides metadata on the .ops attribute of the data that you generate. So you can use this, so it provides how many cells each sample used, and so on. So you can use these values for like to regress effects or so in these differential expression frameworks.
Have you noticed any limitations in this regard? For example, if if a certain sample has 10 times as many cells as another, are you still able to perform differential expression effectively, or would you then need to somehow subsample the larger sample?
So actually, I've never quantified this, but yeah, it's a good question, definitely. So it's something worth exploring. But it's not something that I've encountered, at least in my data, because usually I have quite balanced samples. But yeah, so definitely that's why we also add these quality control metrics, so that, for example, if you see a sample that comes from only 10 cells, I mean, maybe you have SmartSeq and you get like deeper sequence, so deeper sequencing. And so these profiles are still rich, but yeah, if you're working with 10x, yeah, just 10 cells is rather sparse. So yeah, I don't know if I would trust it. But again, these thresholds are just thresholds. So it's you need to follow a rule of thumb, more or less. Yeah.
Thank you.
Thank you.
More questions? Clarifications?
I see one in the chat. So if may I get your slides? I can share the PDF here, or what's the best option? Like, do you store the slides?
Yeah, I saw.
I don't think I was sent any slides, but happy to host them if you want us to.
Yeah, I mean, I have no problem sharing them. So I don't know if I can attach them here, for example. Yes. So,
yeah, you said also that there is the documentation that you present, right? So, I mean, if anyone wants to, we have posted everywhere, also Twitter and Julep, your GitHub, and I think the information as well.
Let's see. Yes. So I can also share slides here.
Yep.
Access to file restricted by your account admin, though.
But you can share them also on Julep maybe, and we can.
Perfect. Yes.
Yeah.
Yeah.
And if anyone wants, they can contact one of us or something, and we can distribute them.
Yeah, and we can post it on the Zulip issue, and
yeah.
we'll also update notes here.
Perfect.
So I don't see any more questions, but
I had a
thank you so
question.
much. Yeah. Sorry, I thought it was for commenting on that.
So one comment on the last thing on the generation of pseudobulk. I don't know if I could tell you the effect right now or how much I explored it, but in one of those big issues on the best practices, I did explore a little bit, like, does setting the number of cells change the output? And it did seem to. I don't know that I took the time to quantify what that effect was, but it did. My reasoning was, if it introduces any effects, then we should probably control the number of cells used in each pseudobulk. But I would be interested if anyone has explored that more.
So the thing is that in theory, you could, so since you can always regress your covariates, you could include this, so the number of cells whenever you perform differential expression testing. Although, however, I don't think the current PyBasic implementation can use continuous covariates. I don't think so. So that's one of the current limitations of the Python implementation. But yeah, definitely.
And then
yeah, it's better to control for it, I would say.
Yeah, and I think when I was doing these tests, I was doing it in Python and, couldn't, have covariate.
No.
My question, then I had a question about pseudobulking in general, and if you had any thoughts on whether reducing like a population of cells within someone to like a single mean, as opposed to at least like a mean and a variance, so you could describe the distribution better. Is it appropriate? How much do you think is lost here? Or do you have any thoughts on that?
It depends on the biological question that you have at hand. So if you want to see general effects between your cell types, pseudobulking, even just like, classic sum and then applying whatever normalization you want, it's still good enough. However, I would still recommend to go to the meta cell approach to try to explore how the different cell states inside each cell type behave. Because the moment you do pseudobulk, you're actually squashing all this cell state variability that you could have. But yeah, it depends on your biological question, because maybe you are not that interested in identifying like a specific cell state or so. So it really depends.
Thanks.
Thank you.
By the way, just to support the package, I ran it, by the way, and I have 1 million of cells. So it runs. I haven't explored yet what it does, but it runs while we're talking. Since I asked you until now, it has run already. I'm on a server, of course, not locally, but fine.
That's nice.
So yeah. So thank you very much for the talk. It was very good. And I think the feedback was very nice and the discussion. Before you go, I would like to ask that you keep attending every second Tuesday. And if any of you would like also to talk and you have relevant work, you can find us also on Julep or send us an email if you find our emails, and everything is on the website of Julep. And you can send us a small description, a small abstract of what you would like to talk about. And then an available dates. I also recommend to send me already an available date and stuff. And then we can also allocate you and see what's the program and something like that. Also in the next meeting, I will probably have created already a Google form so we can gather some kind of feedback, and if you like it, if you want to join more, what do you would like to see? And maybe there you will also have the opportunity, now that I'm thinking of it, to add some abstract and available dates. I see the potential there in the Google form. So yeah,